So I'd like to introduce Dr. Stenerlöw. He obtained his PhD in radiation biology at Uppsala University, Sweden in 1995. He has been a full professor and head of the unit of biomedical radiation since radiation science since 2010. His research has been focused on biological effects of a heavy charge particle beam and radionuclides, especially formation and repair of clustered damaged sites. Currently, his lab studies DNA repair mechanism, how this could be targeted in tumor therapy, and their role in the developing brain. Today, he's going to present his work on DNA damage and repair in treatment of malignant tumors. Dr. Stenerlöw, when you're ready.
Thank you, Behnaz, for this initiation, and I thank also Shirin for inviting me to hold this presentation. I come from the Uppsala University and at the Department of Immunology, Genetics and Pathology. We are placed in a campus at the university called Rudbeck Laboratory. That is a research facility for cancer research, mainly cancer and genetic diseases. And it's located up here in the top of this image. Here is the new entrance building, but we have been here for 20 years or so. And the idea was to have a translational research coming from basic research into to clinic. And we are close to the Uppsala University Hospital. I don't know, can you see my arrow here?
Yes.
Yes, great. So this is Uppsala University Hospital, and we are connected through tunnels to this. And here is actually the Department of Pathology is up here, so, but here we are hosted. Actually, you can see here is the new, this image is some 2-3 years old. Here they started to build the new radiation therapy unit and also the nuclear medicine facilities, the new ones. So they are now up and running. We also have down here, you see the Scandion Clinic. It's a proton therapy facility that is dedicated for proton therapy that has been up and running since 2016. So we're in a very good environment. So what I will talk about today is, the topic is DNA damage and repair in treatment of malignant tumors. So my research has been focused on DNA damage and repair since I started as a PhD student. So that is the basis of the research, and we have translated that into preclinical but in vivo studies, then how we can sensitize tumor cells and tumors in mice models. And also in the later years, we have been focusing also what are the adverse effects, especially when we talk about radiation therapy in treating CNS tumors in children. So I will go through these. I will spend maybe half of my presentation with the DNA damage and repair, and then split the rest between these two topics. So I think basic DNA damage and repair studies has a very important role because there are a lot of things we don't understand when it goes from the initial DNA damage through pathways. There are sensors, transducer of signals. We have pathways that are activated or inhibited. And finally, after a lot of a cascade of events going on, we have DNA repair that tries to do the work and repair these lesions that were induced. So this is now radiation biology course for you that had the exam today. I mean, these are from textbooks. We have a lot of different DNA lesions that are induced by ionizing radiation. I focus on three of these here. The three are the most important I consider. These are base damages, where we have this sugar phosphate backbone is still intact, but the base is damaged and has to be replaced and removed in order to have a functional genome. About 2,000 of these base damages are induced after a typical tumor fraction dose of 2 gray. We have single-strand breaks where the sugar phosphate backbone is damaged on one of the bi-stranded strands on the DNA double helix. About 2,000 of these are also induced after the 2 gray fraction. And finally, we have the double-strand break, where we have on opposite side of the bi-stranded DNA, about 50 of these are produced, and that is a time zero after irradiation exposure without any repair going on. And what one can consider is that the two top of these, the base lesions and the single-strand breaks, they are not very much unique for the cell to encounter. Every day, tens of thousands of these more simple lesions are induced in each single cell due to metabolic processes, replication errors, heat instability, and so on. So we have very effective repair pathways taking care of these. Otherwise, we wouldn't sit here, because without these effective pathways, the cells wouldn't be able to develop and maintain the genomic stability. So these are critical, but not unique. So adding a few thousand of these after irradiation dose of few gray doesn't make a big difference for the cell. So it can easily handle these and repair these fast. And that is done by base excision repair, nucleotide excision repair, and mismatch repair, and some other related pathways. And actually in 2015, the founders of these repair pathways were awarded by the Nobel Prize in 2015. Just pointing out how important these pathways are. There is more troublesome with the DNA double-strand breaks, the DSBs, as you might understand. And we have been interested in these for many years, and how these are, to start with, what do we measure? I mean, everything we do is depending on assays. And we have assays that measure something, but what do they measure? We measure a break when the DNA has been split into two parts, but that could be very different if we use a toxic drug that induces a very simple DNA double-strand break compared to ionizing radiation that could produce more complex DNA double-strand breaks. And another issue is how far do these actually double-strand break is two opposite single-strand breaks. One single-strand break there and one single-strand break. But it could also be splitted and separated by some distance here. One single-strand break there and one there. And we don't know how far can these be in order to be treated as or for the cell to feel it as a double-strand break. We might also have some other lesions, base damages and so on, nearby, adding to the complexity of this damage. So clustered double-strand breaks are induced mainly by high LET radiation, but also actually by X-rays and normal standard low LET radiation. We're very much interested in defects in the repair of these double-strand breaks and pathways activated. And what happens when if we block one pathway, does another one take over and do the work or not? Chromatin compaction, or the organization of chromatin around histones, and we have euchromatin and heterochromatin. These are issues that are very little is known about these things when it comes to repair, production and repairing of DNA double-strand breaks. So, I mean, these are the issues that we are interested in, and some of these I will present today. There is a close link, of course, between DNA double-strand breaks and cell survival and late effects, including the cancer development, although that is taking a lot of time. I have to say also there are some drugs that also induce DNA double-strand breaks that we use as a model of radiation in our research as well. So to start with, I mean, we have to think of how do we detect DNA double-strand breaks, and from where do we get the information? And there are several assays around how we can measure DNA double-strand breaks. One assay or methodology that have been the dominating one in the last 10, 15 years is the so-called foci analysis, where we use immunohistochemistry to detect chromatin modification nearby or near the DNA double-strand break. And we call these surrogate markers because they don't mark the double-strand break, they mark something activated nearby the DNA double-strand break. And one of these protein is the gamma H2AX, it's a phosphorylated H2AX protein. And another one that is very common is 53BP1, that is recognizing modified chromatin. So if we have a DNA double-strand break, we have a lot of chromatin modifications, not only in the close vicinity of the double-strand break, quite far away, up to hundreds of kilo base pairs, up to maybe millions of base pairs from the DSP site. And that helps us because if it wasn't a very strong signal enhancement here, we wouldn't be able to detect these. And what we can do then is we can use antibodies that are labeled by a fluorescent dye that binds to these modifications in the chromatin, and we can detect these as spots or foci. So each foci here or focus here represent one double-strand break, and this cell was irradiated by 25 or 1 gray of X-rays. So we have about 25 to 30 of these spots or foci. And the big advantage with this method is that you can use it in single cell. You can detect clinically relevant dose or responses in the dose of 0 to 2 gray. You can also combine with other protein markers that binds to these that are working in the repair pathways or something like that. So this is just a surrogate marker for the DNA double-strand break. It doesn't show any repair going on or anything like that. And what we see here as a red dot is not 2 proteins or 3 or 4, it's hundreds or thousands of protein molecules lightening up and giving this signal in the microscope. Another previously commonly used assay to detect DNA double-strand break is the pulse field gel electrophoresis, PFGE. It detects fragments of DNA. So when we irradiate cells, the DNA breaks randomly. So like cutting in blind a long string, we get short DNA strands and long DNA strands. So here and then we put these, take away all the proteins in the cell. Each of the base of the DNA is negatively charged. That means if we put it in an electric field and with this pulse field, we put it in different direction, we can have the DNA to migrate. And here we have in the mega base pair size. So we can separate up to 5 to 10 millions of base pair size DNA. So in the control untreated cells, we don't have any small DNA fragments because it hasn't been damaged. So everything stays here in the starting point. But the higher dose we get, the more fragments become smaller and smaller. So we get more and more DNA running fast in this electrophoresis towards the cathode here, or anode, sorry, the plus here. So here we use DNA from thousands of cells. It's not that sensitive as the foci assay, but it's very good in quantitating breaks induced and after some repair. And it's the best one if we want to score the kinetics of the repair. A variant of this is called the COMET assay. It's a single cell electrophoresis that is used in some times, but I will not go into detail about this. So still some basics here, background, and I think many of you are familiar with this, that normal cells, even if this is quite unfamiliar type of damage, the DNA double-strand break, sorry. This cell has a number of the different response or repair systems that could deal with these DNA double-strand breaks. The most important one is a non-homologous N joining, NHA. The alternative repair of double-strand break is homologous recombination. I want to talk about this here. So at time 0, when we have irradiated the cells, they have a lot of broken DNA, different sizes, small fragments, large fragments. As soon as we leave them for repair at 37, they will repair, and after 5-6 hours, we essentially have no breaks left. So it managed to repair these, all the DNA pieces together again. And this is taken care of by this NHA repair pathway, where we have big protein molecules that initially bind to the broken DNA ends here and grab these. There is some trimming. This is a simplification. There are a lot of other proteins involved here. And finally, it's ligated at the end here. So the thing is that this sequence that is left here after repair, after some hour, might not be the same as the DNA sequence as it was before it was irradiated. So there might be some short deletions of a few base pairs or up to maybe 20 base pairs. But it's nothing that normally kills the cell. So this seems to be a rescue pathway for a cell that leaves it to survive by doing this quite quickly and not really accurate. Of course, there are interests then to modify what happens if we can block this. That would be an excellent way to enhance radiotherapy or chemotherapy where radio or DNA damaging agents. So what happens if we block this complex, the DNA PK complex, that is the initial one that grabs the DNA and then further on process the repair. If we block that, there will be no or little repair at all. So even after 5-6 hours of repair, we have still the same fragmentation as we had in the beginning. And if we use this foci assay, we still see more or less the same number of dots per cell as we did before the repair started. So of course, this is a very interesting way if we could use that as a further enhanced radiotherapy, where we normally in wild types are these that have normal repair, they repair within a few hours. These cells that have been blocked by either a drug that blocks this complex DNA PK, or if we can knock it out by using gene knocking out system, they don't repair at all. And we can look at survival here. It's a huge difference. Wild type, this is a typical survival curve for a wild type cell. And these are extremely sensitive with a survival at 2 gray less than 10%, maybe 4 or 5%. Of course, this is, as I said, very attractive, and a lot of drugs are being developed in trying to block this complex and maybe later complex as well. But this will not work if we can't make it specific for the tumor cells. We just save some power in the accelerators because we need just less radiation dose. If we can't make this knockdown or blocking specific for the tumor cells, so we need either to make the drug going to the tumor cells, or use some, if the tumor cells overexpress this protein, that will enhance the effect in the tumor cells. Otherwise, it will not work. So there are a lot of things to do still in this topic. I want to stress this now. I will stress it maybe later on. I mean, there is a need for new assays to detect DNA damage, and specifically DNA double-strand breaks. And we have tried to do that in a number of studies in the last decades. Because the current methods are not really enough. They don't tell us the whole story. And before I go on, I would just tell some thoughts for you that are familiar with the foci assay, where we measure these dots, and that is, for example, gamma HX, that is a phosphorylation. At time 0, just when we irradiate, we don't see anything, because to have this activation of the proteins to form these spots in the cell nucleus, it takes some time. So it takes 5 to 10 minutes to peak, and then not much happened in the first half an hour up to an hour. And then we can see that when the cell have been repairing, the number of these dots that are surrogate markers for double-strand breaks, they are disappearing. Whereas if we use an electrophoretic assay, where we just measure the number of fragments and how much these fragments are rejoined back and repaired back again, we see a much quicker response. So already in 15 minutes, half of the breaks have been rejoined. And after 4 or 5 hours, they are not, everyone more or less are repaired. So there is a mismatch among methodologies, what we can see and how to interpret this. And this is very important to know. And why this is so slow is that it's a biochemical process involving phosphorylation, dephosphorylation, and deactivation of proteins. Therefore, there is a delay compared to when we look at the true double-strand break repair and rejoin. So one should keep that in mind, that what the methodologies show us is these have advantages and disadvantages. To further stress that, when we go to and look into cluster damage sites, as we see after highly irradiation, for example, alpha particles or heavy ions, compared to X-rays, photons, or electrons, where we have a typical survival curve like this, we see that we have a much deeper and more or less exponential dose response when we use highly radiation. And to explain this, or we talk about the relative biological effect, RBE, and trying to investigate what's going on. If we look into, for example, how many double-strand breaks that are induced for a certain dose of highly radiation compared to low radiation, it's not very much different at time 0 before the repair starts. But if we look when the repair goes on for some time, here is a lowly irradiated cells, gamma or X-ray, then most of the cells have repaired their double-strand breaks within a few hours. But if we you look at the same type of repair in highly irradiated cells, in this case it was heavy ions, 125 kV per micrometer ions, then the repair is slower, and we have a high fraction of unrepaired breaks after 4 to 24 hours. The reason why we have this, that is not really known. There have been a lot of theory about this, but no one have really showed clear evidences that there are differences in the type. It could be differences in the complexity of the break, or other differences that makes it much different or more difficult for the cell to repair. And I will show you our contribution to this field, how we try to figure out what is going on. We use, again, the pulse field electrophoresis, and we just not only use one type of separation, we separate on different gels, so we can separate from a range of from 10 kilo base pairs, 10,000 base pairs of the DNA, up to 10 million base pairs of DNA. And then one can plot the distribution of fragments according to the fragment sizes. So we have small fragment sizes here, it's a log scale, and here we have tens of mega base pair sizes. And here is the fraction of the DNA in each size bin. And if we plot these, it's more or less a random distribution if we radiate with photons or X-rays. So they behave as expected, as we have a random breakage. What we and others saw was that if we radiated with highly radiation, we have a deviation from this random distribution. So we have a deficit of large fragments, and we have an excess of small fragments that are not visible normally when we do the normal type of analysis. So we don't look at the very small fragments. So in mass, these are very small, and therefore we can't detect them with normal assays. But if we look at the numbers and count the numbers, there are a lot of these. So we have an excess of small fragments that are normally missed in standard assays. So that again pinpoints the importance of the methodology. And the theory behind this is if we have a ion track or an alpha particle going through the folded chromatin, the chromatin is normally folded in chromatin loops like this. There is a relatively high probability that we have multiple breaks within one or two other loops here, leading to these or release of these small fragments. So by taking these two into account, we can see that we have doubled the number of double-strand breaks that we measure compared to the traditional methods. We also found that, and that is an indirect proof that it's the chromatin folding that is responsible for this release of very small fragments, where we see in intact cells that has an intact chromatin conformation. If we release that and just take away all the proteins, and then we have more or less a random distribution of all the DNA, and then this excess of DNA fragments, or small DNA fragments, more or less disappear, and it goes more or less and follows the random distribution curve as we see for X-rays, indirectly proving that it's the chromatin conformation, the folding here in higher order structures that is responsible for this excess of small fragments. And this is, of course, very important for our understanding. How should we interpret the data if we want to model, for example, what is going on? For modeling is super important to understand how many breaks are induced and where are they induced and how are they repaired. We have also continued with heavy ions and irradiated cells with different LAT. So actually the ion comes here with high energy, it slows down and get lower energy, and as it slows down, the LAT goes from intermediate high up to very high. And here we have used these surrogate markers, gamma HTX and 53BP1 after 5 minutes after irradiation. And we can see how they co-localize. The red and green becomes yellow, then they come in the same spot. So these two proteins perfectly goes into the same spot. So we irradiated these from the side and just can follow each track. So this represents a track of an ion. What turned out was that when we compare this to other assays in quantifying the number of breaks, is that here for this LAT, each spot, even though it looks more or less the same as one spot here, more or less looks the same as one spot in these, although the LAT differs by a factor of 4, these spots here, or foci, on average contain 1 double-strand break, whereas these spots or foci contains on average 3 to 4 double-strand breaks, but we can't see the difference. And that is a problem. And we are really eager to know what's going on in these foci. We have some ideas how to look into that and what is going on and what is the complexity and the DNA small fragmentation doing to these, and how can we detect this and understand this further. So that is an ongoing work in our lab and other labs. So to summarize the first part here about DNA damage and repair, we have large fractions, small fragments induced by high lethal radiation. Clustering may lead to underestimation of the initial number by a factor 2 more. And this clustering is mainly due to the chromatin folding. We know that high lethal radiation slows down the repair, but it's not extremely slow as it has been shown before when we used in these new assays. And also single foci detected by these immunohistochemistry methods after high lethal radiation might contain 1 to 4 or even more double-strand breaks. That makes it very difficult to interpret if we don't have more knowledge about these. So I think to fully understand the formation of DNA double-strand breaks and the repair, we have to use different types of assays. They can tell different things, and for sure we need to develop new ones. So the second part, I will show some examples of what we have been doing, trying to translate, sorry, I have a highly track just over my, let's see, I will try to move. So we have to try to translate our knowledge about DNA repair into a project where we want to block the so-called heat shock protein. Some of you might be familiar with that. That has been ongoing for decades to find inhibitors against this heat shock protein 90. This was discovered a long time ago, as I said, due to heat response, it was activated, but we don't heat the cells here or something like that. So that is just the name that is related to heat. What this protein do, the heat shock protein 90, that's present, it's a huge role in all normal cells and tumor cells. It deals with folding and assembly and translocation of and degradation of proteins. So many proteins, they are just there for a while, and then they are degraded and put into amino acids for later use. And the heat shock proteins are essential here for this folding of the proteins and also the degradation of the proteins. If the heat shock protein 90 is not there, there will be degradation of proteins that shouldn't be there, for example. HSP90 is overexpressed in different types of cancer. And many of the client proteins, there are hundreds of client proteins. Client proteins here means proteins that are interacting, that HSP90 interacts with. So a lot of these are involved in cancer development or tumor development, tissue invasion, in sensitivity to anti-growth proteins, for example, and other growth signals, replication potential, telomerases, apoptosis, for example, cellular metabolism, angiogenesis, and also some of them are related or involved in DNA repair. So we started some years back where we were in contact with a company called Aztecs. So that was some 6-7 years ago now. They had developed a new type of HSP90 inhibitor that was a quite small molecule. And in contrast to the previous one that has quite high liver toxicity, this had 8013, that's as we call it, or Onalespib, that has improved pharmaceutical properties and soluble in water and much less toxic for the cells and animals. It's now in phase 2 clinical studies for prostate cancer and refractory gastrointestinal stromal tumors and so on. And it has also been as a sole agent without any radiation or something like that. And it's also in local regional advanced head and neck cancers after radiotherapy or in combination with cisplatin. So it's ongoing in clinical studies. But when we started this, nothing was done in models of tumors in vivo. So the aim of this when we started was to have characterization in vitro using cell models of this HSP90 inhibitor, explore optimized combinations. So when should we treat with radiation compared to when should we treat with a drug? So what should be the frequency and timing of this? And we try to characterize the biomarkers and traces that we did in vivo as well with using PET imaging and SPECT imaging. And then we went into in vivo study where we had implanted tumors and looked at the tumor response in this. So what one can say is that what we saw in general of the molecular mechanism is that this, when we analyze the cell cycle, so even when we just irradiate, the cell will stop in the cell cycle and try to repair before it goes on. What many drugs do, and especially in combination with ionizing radiation, is that it blocks the cell in the cell cycle. So what we got when we just treated with the drug by itself, without any radiation, we have an accumulation we have G1 here, G2, and S. And here we see G1 is looking as normal, but we have a big accumulation in S phase or late S G2M phase. So sorry, this is G1, S phase, and G2M. So G1, S is in between here and here we have G2M, and then the cell goes on another turn into G1. And in all of these cells, it really increases the fraction of G2M cells. And accumulation in G2M is normally an indication that there are severe deficiency, either in the that we have serious DNA damage, or that are the other proteins transferring these cells from G2 to M and go through or let the cell pass in the mitosis. That is the critical event, going to the next round in the cell cycle. And if we have an accumulation, that is indicating serious problems. One other thing with this is that if we have accumulation of G2M cells, we know that late G2 M and M cells, they're extremely sensitive to ionizing radiation. So if we, after exposure to this drug, then go on with radiation, then we have extremely sensitive cells compared to in other cell cycle phases. So I will not go into detail. We looked into a lot of proteins here, exposing these to drugs of different concentrations. We can just look at this. HSP90, it's downregulated. We have some of the repair proteins here are downregulated. We have the epidermal growth factor that is very important in tumor growth. That is also downregulated, and some other proteins are downregulated, whereas some are not affected at all. So some proteins are affected and some are not, as we expected. And some of these are very important for tumor growth and the repairability after radiation therapy. We also saw that there was, when we combined the radiation and the 8013, the Onalespib, that is a HSP90 inhibitor, we saw that we had an increase in the number of cells with a large number of foci per cell. So there is some effect on the repair, although it's not exactly clear how important this is. We looked different cell lines here. There are 4 different cell lines. We have different concentrations of the Onalespib and the radiation, and we see that we have a sensitization, and we found that there is a synergistic effect that is very promising in these monolayer cultures as these are. So we went on using a tumor model called spheroid. So we call it spheroid. It's a 3D tumor cell culture. So this is normally monolayer that grows on one, 2-dimensional in the cell flask. If we take these cells and put them into specifically treated well, that it's very slippery, so they can't attach or adhere to this surface. After a while, they find out, okay, it's better we stay together. So they form and start to grow in a sphere like this. And this grows and grows and grows. And this is a little bit more complex tumor model than it is if we just use monolayers. So what we do is that we produce these tumor spheres, and then we start a treatment, and then we can follow how these spheres are growing or not growing. And we see that there is a clear decline or complete block actually of growth if we combine radiation and a drug in higher concentrations. So we can clearly see here is a control with sphere grows and become parts of a millimeter big, and here it's much less grow after this treatment, combination with 5 times 2 gray only and a small concentration or low concentration of this drug. We went further then, so we implant tumors in the hind of the mice. I can use different tumor types in different sides of the animal. In this case, we just had on one side, and we did the same. We had a treatment with the with the HSP9 inhibitor in combination with radiation. For ethical reasons, we just run on 3 treatment days. So that is a very short therapeutic schedule, if one can say so. So we just have 3 days after the tumor has formed, and then we follow the tumor growth with time. And we see that when we have a combination, the high drug concentration together with radiation, it's almost possible to completely block the growth of the tumor, although we just have a very short treatment schedule. And here one can plot the survival of the animals. So here we have the control that has a maximum lifespan or average mean survival of 9.5 days control, compared to the high dose here, 10 milligram plus radiation that has 31 days. I can skip this. This is the tissue from the tumors. So to summarize the part 2, this drug is very promising. It has a very low IC50. It downregulates client proteins that we know are important for tumor growth. It's radiosensitizing. It has synergistic effects, increases the apoptosis and cell cycle arrest. And this is also in line with in vivo studies. It goes in line completely with what we see in vitro, and that is a promising, very promising results. And we have published some of this work in recent years. So, to my last project, started actually, we were contacted by toxicologists that were interested to see that had tested how different toxic and environmental agents affect the brain development and leads to leads to cognitive defects after exposure at young age. And this, of course, have a relevance for humans, but we use mice, of course. For example, CT investigations of the brain, quite high doses, 30 to 50 milligray. And then we have, of course, radiation therapy of brain tumors. Their very high doses are used. What we know is that although the mature brain is quite insensitive to radiation, the developing brain in children is very sensitive when it comes to disturbances leading to potential cognitive defects later on. So if we have a young exposure, we know there is, and that is known for tumor therapy, that they suffer from late cognitive impairment later on. What we don't know about yet is that if this alters also the susceptibility to other treatments later on when they grow up, or if it accelerates the aging and other related issues. So these are important things we would like to put forward later on. So the brain, we were are very interested in this and how it relates then to ionizing radiation. As I said, the brain development is very highly sensitive to everything that that disturbs this. And what we have been interested in is how ionizing radiation by itself, but also how it might interact with common chemicals or pharmaceuticals that are used in children in the clinic. So what we use is that we use mice. We expose them at an early age they are 10 days old. So this is called a neonatal period. They're 10 days old. And then we wait, and then we see then they are grown up. They are 2 to 4 months. And then we see how they perform in the behavioral test to study cognitive function. And why we use the 10-day-old mice is that mice here has what is called the brain growth sprout. That's when the growth of the brain explodes, not in number of cells, but in number of connection between neural cells. So the number of cells are not really changing very much, but the connections. So one cell could be connected to hundreds of thousands of different other cells. So that is building up our neural network. So that takes place in the peak 10 days after the mice is born. In humans, it's slightly different. Then the brain development is very active at the last trimester, and then it continues, although it declines after the birth of the child, but continues up to maybe 20, 30 years of age. But this period of time, the first 5 years in humans, are known to be very sensitive period. So for mice, then we have a critical window of PD10, that is postnatal dead 10 days after birth. That gives us a very interesting or very sensitive model system if we can expose mice at this stage. So what one can do then, how to study behavior in mice. In humans, we can ask questions, they can perform a lot of things. That can also be done on mice. We can make them swim, we can let them try to find and solve tasks and so on. But one of the most sensitive ones developed by Per Eriksson is how they behave in a normal environment. So what one do is one have cages that look as the cage they are familiar with. So mice are very, they don't want to be disturbed. They want to feel very comfortable with the environment. So then one put them in a cage that looks like the home environment. The only difference is that it has a lot of sensors, so it can measure how they move in that direction and up and down. So we expose the mice at 10 days after birth and then measure the cognitive function at 2 to 4 months after. So what one can see when one take out the mice or move the mice from the home cage into this test cage, then in the beginning, they run a lot in the beginning. So in the first 20 minutes, they try to figure out, where am I and what is this? And then they realize after the next 20 minutes period that, okay, this seems to be familiar. There are no threats and so on. And in the next 20 minutes period, they go to rest or go very slowly around and look, and they feel safe. So this is a normal habituation. They get habituated to the new environment. So this is how it should look in the untreated samples or mice. What happens if we irradiate the mice at 10 days after birth is with 0.5 gray here. We see that the control here, the green one, control goes down. They have a lot of activity that goes down to 0. If we use half a gray, it doesn't go down. It starts here and nothing happens. So they go around and look and look and look, and they don't feel comfortable with this new environment. So we saw big difference compared to the control. And also at 0.35 gray, we don't see any big difference here, but we can see that they have lower activity in the beginning, and this has even lower activity. That is also indicating some disturbances in the cognitive function. I will skip this. We have looked into proteins, what are activated and so on. I will not go into that. What we have been interested in the last couple of years is that if pharmaceuticals can interact with ionized radiation and worsen the effect of the radiotherapy by itself. And we have been using ketamine as a model agent for anesthetics and sedatives, combined with low doses of ionized radiation. So we have a neonatal exposure at the 10 to 15 days after birth to radiation and pharmaceuticals. And then we have early events. The first week we score DNA damage and repair in tissue and so on. And then we look at behavior and protein gene expression, and then they become old. So to summarize what we have seen, we have done a lot of this and a lot of different types of drugs in combination with radiation and we can focus on the control here, the green. In the beginning, they are quite active, less active, and after the last test period, they are going to sleep more or less. Whereas if we have 50 milligray, 100 milligray, 200 milligray, it's the same. These are the ketamine alone, it goes down. 50 milligray, 100 milligray, 200 milligray alone. These three here, they go down and nothing happens. So low doses below 300-400 milligray and low doses of ketamine here. And this ketamine dose is so low, so they're not sleeping, they're just a little bit slow. They don't give any effect by themselves. But if we combine 50 milligray, if we combine 100 milligray with ketamine or 200 milligray with ketamine, we see that it doesn't go down. So here we see still after this last 20 minutes of test period, they still are not comfortable and they are running around and have some disturbance in the cognitive function. And this has been confirmed by other tests as well. And what we saw also, that is very interesting, that in these that have been treated with ketamine and radiation together, they have not less neural growth out, they have more growth of the dendrites in the neurons. So we have more dendrites and they are longer in these groups compared to the controls and the other treatment groups. And that is something we are looking into now for further analysis. So summary, the last summary is that about at 0.4 gray, we have persistent alternations in cognitive function in mice. If it's exposed at a low age, in mature mice, we don't see anything. We have molecular changes in the adult mice. We also see that ionizing radiation can interact with chemicals and pharmaceuticals and give cognitive defects even at 0.1 gray. And this means that we shift the dose response from 0.4 to less than 0.1 gray if we combine with pharmaceuticals that are commonly used at the hospital. We have changes in neural structure, and ongoing we look at protective agents and other pharmaceuticals and mechanisms. So by saying that, that was my last slide. I just want to thank you. And here are some other collaborators at Uppsala University, at Rome and Helmut Center in Munich. And with support from the Swedish Cancer Society, Childhood Cancer Society, and Radiation Protection Institute. And we had an EU project that's now run out. And thanks for listening. And a normal Valborg, the last of April, that's today, it will look like this downtown Uppsala. About 150,000 people are running around there and celebrating the spring coming to Uppsala, but not this year and not last year. So thanks.